Hello everyone, welcome to Exam Essentials, and today we'll be solving this Biology IGCSE Paper 2 from November 2024 examination series. So without wasting any more time, let's just get into it. The first question is about a passage, and we are going to read this passage and understand it along way, and apart from that, we'll be solving the questions of it as well. So read the passage below. Use information in the passage and your own knowledge to answer the questions that follow. Kidney transplants. So what does this transplant mean? Transplant basically means to transfer something such as an organ from one person to the other or from one animal to the other. This is the basic definition of transplant. The main role of kidneys is to filter waste products from the blood. If the kidneys lose this ability, waste products can build up, which could lead to death. This loss of kidney function, known as kidney failure, is the most common reason for needing a kidney transplant. So basically, you have to take a kidney from one person, right, to replace your damaged one. And this is what we refer to as kidney transplant. And your blood builds up waste products, right? As it passes from one cell to the other. So your blood basically builds up this waste products. Now your kidney is needed to filter these waste products. Now this kidney is needed to filter this waste product. If this waste product is not filtered, then the blood can become toxic and that is going to be lethal for your health as well. So yeah, it is possible to use kidney machine to partially perform the functions of the kidney, a blood filtering procedure known as dialysis. However, this can be inconvenient and it takes a long time. A kidney transplant is a better solution for kidney failure whenever possible. Okay, so basically you can artificially filter your blood using a machine that is what we refer to as a blood filtering machine, and that procedure is known as dialysis. I mean, this thing is going to work same as the kidneys itself, but this is going to be highly inconvenient as you have to get to hospital every week, or I mean, yeah, you have to get to hospital every week to replace your blood, right? And that is going to be extremely inconvenient for some of us. And yeah, that is one of the main reasons that this dialysis procedure is inconvenient, as you have to visit the hospital quite often. And yeah, apart from that, it takes up time to do this procedure. So this is really inconvenient process. So this was, I mean, remember that this was the cons of this process. I have already said some of the cons of this process. Sorry, guys. A kidney transplant is suitable for people of all ages, okay, who needs to have one, if they are fit enough for a surgery, okay. If a patient has an infection, this will need to be cured before the transplant, okay. The person who receives the transplant must agree to continue with the recommended treatment. So after the transplant has been done, the person needs to have some medicines or the person needs to undergo some treatments. After the transplant, these treatments would include medication known as immunosuppressant drugs. So basically, every body cell of ours has these markers, on its cell, right? And these markers is what we refer to as antigen. Now, what is the main purpose of this antigen? Now, we have immune cells, right? Like the white blood cells, WBC. Let me write this for short. Immune cells, white blood cell, right? And white blood cell comprises of lymphocytes and phagocytes, right? Now, what are the main role of these lymphocytes and phagocytes? They are responsible for identifying foreign cells and destroying it, right? Now, how does this lymphocyte and phagocyte identifies some foreign cell, right? This is because of the cell marker, that is antigen. So the antigens of our body cell is going to be different than the antigen of this pathogen or something like that, right? The antigen of that new kidney that has been taken from a different patient, right? Now, the main purpose of this immunosuppressant drug is to weaken the immune system so that it couldn't show up this immune response, and hence yeah, this would lead to the body to accept the kidney, the new kidney that has been transplanted. People have two kidneys, but they can survive well with only one of it. This means that it is possible for a person to donate a kidney while they are still alive. This is known as a living donation. So whenever you donate a kidney, whenever you are alive, this is what we refer to as a living donation. Clear? People who want to be considered as a living donor are carefully tested to make sure that they are a suitable donor and are fit for operation needed to remove a kidney. Living donations are based from uh, living donations are based from a close relative, right? Because they are gonna have similar antigen when compared with the patient. As this reduces the risk of body rejecting the kidney. In the United States, the number of living donation in year 2020 was 1040, and in 2021, the number of living donation was 429, right? So this was the number of kidney transplant that has taken place in the year 2020 and 2021, right? So these are basically datas, and they may ask you, I have marked this, right? So whenever you come across these datas, just mark it down because they may ask you to find the percentage increase or percentage decrease in the years between 2020 and 2021, right? You never know. So basically, it would be better for you if you mark all of this along with I have done that, and after I have done all of this, I have solved up the question paper. I've solved up the entire question paper. So basically, people would go on watching up the mark screen and solving the question papers, but I really do not follow that pattern, to be honest. I would assess myself before doing up this before taking up any classes of mine. So what I do is um, I did up the paper on my own, and after I was done with this paper, I then cross-checked it with the mark scheme, right? And that is how you should be doing each and every paper that you are doing at this point from now onwards. Because yeah, that is going to help you. That is going to self-assess you. After you assess yourself, you're gonna identify some of the problems. And when you have these problems, what you're gonna do? You're gonna read the book, use GPT, or even ask your teacher and so on, right? So yeah, that is the ultimate thing that you should be doing at this point, because you have like 4 months approximately left for your exams, right? And that is how you should be preparing before your exam from now onwards. And if you're facing any problems with biology IGCSE, then make sure to comment below. I would be more than glad to help you all. Okay, so moving on. Kidney donations are also possible from people who have recently died, okay? First of all, remember that there was this living donation, and now they say that kidney donation can also be possible from patients or people that have recently passed away, okay? This is known as deceased kidney donation or passed away kidney donation, and the other one is what we refer to as live kidney donation. Remember that. However, this type of donation has a slightly lower chance of long-term success. In the United Kingdom, the number of deceased donation in 2020 was 2283, 2283 in the year 2020, and in 2021 the number of deceased donation was 1863. Okay, in 2 years, people who need a kidney transplant but do not have a living donor will have to wait until a deceased donor have become available. Okay. In the United Kingdom, the average waiting time for a deceased kidney donor transplant is between 30 months and 36 months. All right. Kidney donors are particularly required from people of non-white ethnic origin, as kidney disease is more common in people of South Asian. Okay. So people in South Asia have this kidney disease, right? They would often have this kidney disease, African and Afro-Caribbean ethnic origin. However, there are not many donors from this origin. So basically, people from the South Asian, African, and Afro-Caribbean ethnic group would have a greater proportion of kidney diseases. However, there aren't many donors from these communities or from these regions, right? So that is one of the point. If I was you, I would be marking this, right? Because a question may pop up, right? So um, when a person receives a kidney from a living donor, this is a carefully planned operation. If they are waiting for a deceased kidney donor, they will be contacted when a suitable kidney becomes available. Okay. The patient that has surgery to insert the new kidney, and the patient then has surgery to insert the new kidney and connect it to their blood vessels and bladder. Okay. The new kidney will be placed in the lower part of the abdomen. The patient's own failed kidney will usually be left in the place. Okay, so um, your unfunctional your your kidney that is not functional will be kept in place, right? And then you'll be inserted with a new kidney, right? So yeah, that is what they've said over here. So state two products that sorry guys, state two products that kidney removes from the blood. So what are the two products that kidney removes from the blood? So first of all, this is going to be urea, right? So this is going to be urea, and the other thing is going to be water, right? So yeah, this is going to be dialysis can be inconvenient and takes a long time. Suggest two other reason why a kidney transplant is a better solution uh, for kidney failure than dialysis. So as I said that the person does not need to visit the hospital again and again could be one of the reason, and the other reason is that this would lead to better success. This would lead to better success, right? Or efficient filtration or something like this. So does not need the patient to visit the hospital repeatedly, and the patient could lead a normal life, right? Because the patient does not need to visit the hospital again and again, and the patient could basically go on to its normal life and do exercises and carry out his jobs and anything, right? So yeah, there won't be any tensions of any sort. So that is what I've said over here. So first thing is that the patient doesn't need to visit the hospital repeatedly, and the second thing is that the patient can lead a normal life. Moving on. The patient receiving the transplant must take immunosuppressant drugs after the operation. These drugs reduce the action of the immune system, right? So describe how immune system responds to a disease, right? So basically, suppose that this is a pathogen, right? And there are these antigens, as I said. Antigens are cell markers that could be found on cells. These are antigens, right? Now our white blood cells cannot recognize this. And now our white blood cell cannot recognize these pathogens, right? And lymphocytes, and we have a type of white blood cell that is known as lymphocytes. And lymphocytes are responsible for producing antibodies. Now, these antibodies are a type of protein that binds with the antigen of this pathogen, right? That binds with the antigens of this pathogen. And once it binds with the antigen of the pathogen, it causes the pathogen to stick together. And these antibodies acts as a label, and these antibodies then acts as a label, right? And these antibodies then acts as a label, right? So it acts as an identifier. It acts as an identifier so that the phagocytes, there is this another type of white blood cell known as phagocytes, so that this phagocyte could quickly identify this pathogen and engulf it by the process of phagocytosis and destroy it, right? So this is what I've written over here. The white blood cell recognizes the foreign antigens on the pathogen. Phagocyte then engulfs the pathogen by the process of phagocytosis and destroys it. Lymphocytes are responsible for producing antibodies and memory cells. Moving on to the next part of the question. Explain why the chances of rejection are lower if the kidney comes from a close relative, right? So if the kidney comes from a close relative, it is gonna have similar antigen. It is gonna have similar antigen as the patient or the person receiving up the kidney, right? And that is gonna decrease up the immune response. Kidney coming from close relative has similar antigen as the patient, and as a result, immune response does not take place, or it decreases the immune response. The number of donations between 2020 and 2021, the number of donations changed between 2020 and 2021. calculate the difference between the percentage in living donations from 2020 to 2021 and the percentage change in deceased donation from 2020 to 2021, right? So what basically you have to do is you have to find the percentage difference between 2020 and 2021 for this living one, and you also have to find this percentage change from this deceased donation from 2020 to 2021 non-living, right? And once you have it, what you're going to do is you're going to subtract these two, you're going to difference, you're going to subtract these two and find your answer. So how do you find the percentage change of something, right? Percentage change or percentage increase is going to be final minus initial time by initial times 100. So your initial is going to be the first value, final is going to be the second value, right? So the number of deaths, the number of kidney transplants in the year 2021 is going to be your final, and the initial is going to be 2020, right? And the initial is going to be 2020, right? So then you multiply that with 100 and you get the percentage change in the living donations in the year 2020 to 2021. So that is what I have written over here. So percentage change is final minus initial by initials time 100. And for living in the year 2020 to 2021 is going to be 429 and 1040. Remember, I have marked this. So these data are taken from here. This and this is for living, and this is for this is kidneys, right? I have taken the data from there. So yeah. So yeah, 429 minus 1040 by 1040 times 100 gives us a percentage change of 58.75%. And percentage change in disease is going to be 1836 minus 2283 by 2283 times 100. So that is going to be 19.58%. Now there is going to be this negative sign, right? And you could omit this negative sign. You could omit this negative sign. Now, the percentage difference is going to be bigger value minus the smaller value. That gives us a percentage difference of 39.2%. So yeah, basic math. Moving on. Suggest a reason why there are fewer transplant operations from living donors than from deceased donor, right? So the living donors wouldn't be willing to give their own kidneys when they're alive, as it is scary, obviously, right? And they may be scared that something may go wrong during the operation, and so on, right? Hence, they aren't willing to donate their kidneys, and that is the main reason that there are fewer kidney operation from living donors than from deceased one. People are unwilling or reluctant to donate when they are still alive. Moving on, suggest what is meant by a suitable kidney, right? A suitable kidney is the one that would have similar antigen. So a suitable kidney is the one that would have similar antigen as the patient receiving it, and apart from that, it shouldn't be rejected by the immune system of the body. Kidney from the donor that has similar antigen as the patient and should not be rejected by the body of the person. So explain why kidney donors are particularly required from people of South African Afro-Caribbean ethnic region, right? So first of all, there's as I said over there, if you haven't watched the first part of it, then I would suggest that you watch the part where I was going through the passage itself, right? And then you come to this question. So I have said over there that there's a high proportion of kidney diseases in these regions, and there's a high proportion of kidney disease in the people of these regions, and there are fewer donors over there, right? So yeah, people from these regions are often affected with kidney diseases. However, there are less donors. Moving on. So basically, we're done with question one. Moving on to question two. Some scientists used a calorimeter to compare energy content of different types of grain. This is a calorimeter, and yeah. So let's just go on with the method. First of all, put 20 centimeter cube of water in a calorimeter. So this is the calorimeter, and you put water over here, and the volume is 20 centimeter cube of it. Measure the initial temperature, and that is going to be your T1 using the thermometer, obviously. There's 1 gram of a grain type in a small dish over here. So basically you put a grain sample over here, and the mass of it is obviously going to be 1 gram of it. Now place the small dish in the calorimeter. Okay, you then place it in a calorimeter. Light the grain in that small dish. So basically you're gonna light it up, right? You're gonna catch. Um you're gonna do something so that it catches a fire, right? So basically that is how it is gonna work at this point. So you're gonna light this up, allow the grains to completely burn, because this thing is burning, right? And you're providing oxygen with it, right? So what would happen? A combustion reaction would take place, right? Because when something burns in the presence of oxygen, that is what we refer to as a combustion reaction, right? So this is what we refer to as combustion reaction. And the food over here is acting as a fuel, right? So measure the final temperature. So as this thing burns, this is going to heat up the water, right? So um, gases produced by this or heat produced by this would travel through this twisted pipe, right? As it travels through this twisted pipe, it is gonna cause this water inside this calorimeter to heat up, right? And the heat and the temperature would be detected by this thermometer, right? So you would have your T2, right? And initially you would have T1, right? So to find the difference in temperature, what you're going to do is T2 minus T1. So this is the procedure of this experiment. And they said that they repeat this method with different types of grains, okay? So moving on, explain how scientists could use this measurement to calculate the energy released by 1 gram of the grain, right? So how do you do this? If you have chemistry, by now you should be knowing that you should be using this formula. Q is equals to mc delta T. If you have chemistry, by default, you should be knowing that you should be using formula, but M is the mass of water, specific heat capacity is C, and it has a value of 4.2, and delta T is the change in temperature. Change in temperature, that is going to be T2 minus T1, or final temperature minus initial, right? And how you're going to get the mass of water, provided that you have the volume. So density is going to be mass by volume, right? And you know that the density of water is 1 gram per centimeter cube, and mass, and you have a volume of 20. So the mass of water is going to be 20 grams, right? Now you're going to equate all of these into this formula and find the value of energy released by this grain when one gram of this grain burns, right? So yeah, that is what I have written down in my description, but in a detailed and precise way, obviously. So a volume of water, first of all, needs to be converted into mass, and mass of water should be 20 grams. This is because density equals to mass by volume, and density of water is 1 gram per centimeter cube. Change in temperature needs to be calculated, which is final temperature minus the initial temperature, that is T2 minus T1. Then equate the values in the formula Q equals to mc del T to find the energy released when we equate this in this equation to find the energy released when 1 gram of grain is burned. C is the specific heat capacity, and it has a value of 4.2. So that is how you should describe something, right? You should describe it elaborately, because it no, okay, so yeah, you have to explain, obviously. State how scientists can make sure that the results are reliable, right? So whenever they say something related to reliability, what you have to do is you have to repeat the experiment and find a mean. And this is the only way that you increase the reliability of an experiment, right? Reliability would mean credibility of an experiment. So by repeating the experiment and finding a mean, this calorimeter has a twisted gas pipe passing through water. Explain how twisted gas pipe improves the accuracy of scientists' results compared with a straight pipe. So first of all, as it is twisted, it is going to increase the surface area. It is going to increase what? It is going to increase the surface area when compared with a pipe if it was only a straight line like this, right? So first of all, it is going to increase the surface area. And second of all is that it is going to ensure that the water is uniformly heated. The water is uniformly heated. The water is uniformly heated, right? So yeah, this should be it. Moving on. Twisted pipe ensures that ensures the uniform heating of water when compared with a straight pipe. This is because twisted pipe has a higher surface area than a straight pipe. Moving on, everyone. The colorimeter burns the grain in oxygen. Give a reason why this improves the accuracy of scientist results. So as I said earlier, that this is going to be a combustion reaction, right? So it burns fully. Complete combustion takes place. Moving on. Scientists also collected, okay, this is going to be some sort of a table. Scientists also collected data on the protein, fat, and carbohydrate, on the protein, fat, carbohydrate, and fiber content of the grains. The table shows some of the scientist results. For oat, you have uh, protein in grams per 100. So per 100, so for in 100 grams of oats, you have this 13 grams of protein. In 100 grams of oat, you have 7 grams of fat and so on, right? So these are the values. So these are this, and this is the total energy content. So calculate the mass of protein in 65 gram of rice. Okay, mass of protein in rice. So first of all, what you have to know is that calculate the mass of protein in 65 gram of rice. So in 100 grams of rice, you have 7 grams of protein. How did I understand that? Because it said proteins in gram per 100 gram per. It means divided by something, right? It means that in 100 gram of rice, we have 1 gram of protein. So in 1 gram of rice, we'd have how many grams of protein? And in 65 grams of rice, we will have how many grams of protein, right? So basically unitary math. So as I said, 100 grams of rice has 7 grams of protein. 1 gram of rice would have 7 by 100 grams of protein. And 65 grams of rice would have 7 by 100 times 65 grams of protein. That comes to a value that is 4.55 grams. Moving on to the next part of this question. A long distance athlete wants to choose a grain to add to their diet. Comment on the sustainability of each grain type for the diet of a long distance athlete. Okay. So what what um what grain type the long distance athlete should have in order to run long distance? That is the question. So sustainability, okay, so I could say rice because it has the highest amount of carbohydrate that is needed by the cells of that person to respire, to provide him with energy in the forms of ATP, right, as aerobic respiration takes place. So glucose with oxygen burns on to produce carbon dioxide plus water plus a large amount of energy. So yeah, I could say a comment on this regarding rice. And on oats, I could also say the same thing. It has high amount of carbohydrate and fat. It has high amount of carbohydrate and fat, and it could give energy to the respiring cells of this person. And also, however, there's a disadvantage to this as well. Okay, no, let's just go with this. And also you could say that it has highest amount of total energy, so it could keep the person going on for a long time. And for rye and for rye um and for rye there's this low amount of fat, right? However, there's this amount of carbohydrate as well, right? But let's just go on with this fiber with rye, because we have already did with carbohydrate. So with rye, the amount of fiber is 14, so the person is not gonna have constipation. And for wheat, it has the highest amount of fat, right? And although it could provide energy, but it could also lead to the risks of heart diseases, right? Heart diseases, you could say of this over here. Or it has the lowest percentage of carbohydrates, so it wouldn't give much energy for the person to keep on running. So yeah, that was more or less about this. So first of all, I said that rye has the lowest amount of fat per 100 gram and provides the lowest energy for running. Hence, the athlete cannot run much rye. Then, wheat has the lowest amount of carbohydrate per 100 grams. Hence, the athlete will lack energy for running, right? It has the lowest amount of carbohydrate, and we need this carbohydrate to provide us with a source of glucose that we need to carry out aerobic respiration, and we need some sorts of levels of fat, as I said over here. But excess of it could lead to heart diseases and so on. Oats is the most sustainable at it, as it has both carbohydrate and fat per 100 gram in moderate proportion and provides highest amount of energy for what? For running, obviously. And rice can also be used as it has highest amount of carbohydrate and provides high energy for running. Clear so far? So yeah, moving on to the next question.
Hi, are you an October 2025 O level candidate? Your exams are just 4 months away, but you are still feeling stuck. Still doing classes, watching YouTube, but no real improvement, right? You are not alone, and it's not your fault. It's the method. Let's fix it. Welcome to Exam Essentials, where every second counts and every session delivers. We are launching the High Impact Crash Course October 2025, designed for both Edexcel and Cambridge students. The High Impact Crash Course will start from 10th July and till 13th August. We'll be covering the syllabus in detail for each and every subject. And right after the end of a chapter, you guys will be doing the Exam Essentials Premium Mega Revision Worksheet, which will allow you guys to get familiar with the recent question papers. And in the month of September, we will be doing extensive paper solving, the past papers, from the year June 2019 to October 2025. Now, you guys might be saying, Sir, I face a lot of difficulties whenever I solve a question paper. Don't worry, dear. In the month of October, our teachers will be having one-to-one sessions with each and every one of you guys to solve your personal problems regarding the question paper or even the syllabus. And after the problem solving sessions, you guys will be giving mock exams. And our mock exams, along with the feedback, are absolutely free for our students. And not only this high impact crash course also includes 20 plus question paper solving videos for each and every subject. There will be digital notes, worksheets, and we will be providing you the recorded sessions of each and every live classes that you guys can rewatch anytime. And all the teachers will be available 24 hours, 7 days to clear your doubts. Stop guessing, stop panicking, start learning the way toppers do. Now, you guys might also be thinking, what are the results of our previous students? The results of our previous students are extremely good. They have done very well. So, without wasting any more time, sign up now and gear up for success with the ultimate O level crash course. Message us on our Facebook page Exam Essentials or WhatsApp us at the number provided in the description box, or you can see the WhatsApp number right here. Book your first trial class. Your top grade is still possible. Start today.
Question number 3. The diagram shows how human blood develops from stem cells. So what basically are these stem cells? Stem cells are cells that are undifferentiated, and they could divide into any type of cells, right? Undifferentiated means that they aren't specialized for a particular job, right? They can take the shape and form of any other cells and carry out their function. So the diagram shows how human blood develops from blood stem cells. You have your blood stem cells, and it is dividing into two: myeloid cell and lymphoid cell. Myeloid cell are responsible for producing um, erythrocytes, myoblast, and platelets, right? These are RBC, red blood cell. These are myoblast and these are platelets. Platelets are needed for blood clotting. Myoblast is divided into basophils, neutrophils, Eosinophils and monocyte, right? These are a type of phagocyte. You do not need to have much details on this at this point, but in your A2 exams, in your A2 biology, you'll be having a detailed understanding of this. And these lymph are divided into lymphoblasts that divides into lymphocyte, B, T, and natural killer cell. So basically, again, you do not need to know the function of B, T, lymphocyte, and everything. You just have to know that lymphocytes are responsible for producing antibodies for your level, and you're gonna learn all of these in your A levels, in your A2 exam. Before in your you're gonna need all of these in your A2 syllabus, not now. So describe how stem cells are different from most body cells, right? So stem cells are undifferentiated and can differentiate into any type of cells. Stem cells are undifferentiated and divided by mitosis to form other type of specialized cells of the body. Describe the role of platelets in the body. So whenever, suppose this is your body, right, and this part of it has been cut, or there is a wound around it, right? So as a result, blood could easily pass through this opening, right? So what your platelet does is once platelet gets to this site, platelet gets activated and leads to the conversion of fibrinogen to fibrin, right? And this fibrin forms a mesh around this one. So suppose this is the wound and a fibrin forms a mesh around it. Now, what is the advantage of it? It prevents further loss of the blood. And apart from that, it prevents the entry of microorganisms from the environment inside your blood. So that is the role of platelets, and you're going to have a detailed understanding and even in more depth idea of this in your A-level exams. But for the time being, you just have to know the basic role of platelets. So basically, that's what I have said over here. When a person cuts itself, platelet gets platelets gets to the damaged area and form a mesh-like structure around the wound. This is because platelets gets activated and results in the conversion of fibrinogen to fibrin. This is a soluble protein and this is an insoluble protein that traps red blood cell and prevents the entry of pathogens to your body, right, or to your blood. Moving on, explain how vaccination protects the person from a disease, right? So could find this thing from your chapter 5 of your book, if I'm not wrong, circulatory systems and so on. So what vaccination does is it provides, remember that antigen comment. If you don't, then please make sure to go and take a look at the earlier video as well, right? So the vaccination comment, in that vaccination comment, so sorry, in that pathogen and antigen comment, if you didn't, then make sure to go and check up the earlier parts of this video. Now, the vaccine provides with an antigen. The vaccine provides with a source of this antigen. Source of this antigen is obviously going to be from a weaker strain of the actual bacteria or from a dead bacteria, obviously. But these antigens won't be recognized by our immune cells, right? And our immune cells would result in an immune response, and the lymphocyte would be producing up these antibodies that would bind to this antigen and act as a label for the phagocytes to quickly identify it. And apart from that, memory cells are going to be produced as well. Now, the first exposure of a person to a pathogen is what we refer to as primary exposure. And if the person gets exposed to the same type of pathogen again, or an actual pathogen again, then the person undergoes secondary immune response. And the secondary immune response is going to be faster than the primary immune response, and antibody produced by the secondary immune response is going to be way more faster and more efficient than the primary immune response, right? So vaccine gives you the primary immune response, and if you actually contact that disease, then you're going to have secondary immune response. So that's what I've written over here. Vaccine provides a person with a source of a weaker strain of the actual pathogen. Antigens on the surface of the weaker strain won't be recognized by the immune cells. As a result, antibodies produced by lymphocyte will bind to the pathogen and cause the pathogen to stick together. This results in the pathogen to quickly in the phagocytes to quickly identify the pathogen and destroy it by the process of phagocytosis. Memory cells are also produced. The exposure of a vaccinated person with the same strain of pathogen will result in a secondary immune response and faster production of antibodies. Exposure of the person with the pathogen for the first time is what we refer to as primary immune response. Moving on, body cells can be used to treat blood stem cells can be used to treat blood disorders such as sickle cell anemia, which affects red blood cell and hence would affect, as it affects red blood cell, this is obviously going to affect its ability to carry oxygen, right? And leukemia, which affects white blood cells. So in leukemia, what it does is the white blood cell destroy the white blood cell. It is basically referred to as blood cancer, right? So the white blood cell multiplies uncontrollably. Now, explain why blood cells can be used to treat a variety of disorders. So basically, your blood stem cells are undifferentiated. The same thing repeats again because in your level, you do not have much of it. You just have one more thing, that is, you have just one simple thing, that is, stem cells are undifferentiated and could differentiate into any type of cells by the process of mitosis, right? It could differentiate into this red blood cell that would allow the blood cells to carry oxygen, and it could also divide into white blood cell that would result in an immune response. So that's what I have written over here. Stem cells are undifferentiated and could differentiate into any types of blood cell by the process of mitosis. For an instance, stem cells can make red blood cells that transport oxygen. Stem cells can also make white blood cell that is responsible for destroying the pathogen. Moving on to question number 4. Give me a moment, guys. Sorry for the delay. Diagram 4. The diagram 4 shows a section through a skin with some strict structures labeled, okay? So P, Q, this is your sweat glands, obviously. This is your hair erector muscles. Okay, and this is gonna be your blood vessel. Okay, this is your body here or something. Which structure is the sweat gland? This is obviously gonna be P. Which structure is the capillary? This is gonna be T. Describe the changes that takes place in the blood vessel in the skin if a person moves to a hot environment. Okay, so what are the changes that takes place in the blood vessel? So first of all, you have to know that suppose this is your skin. This is your skin, right? And this is the blood vessel, right? So whenever you're moving to a hot environment, assuming this is the skin, right? And this is the blood vessel, the blood vessel would dilate, right? The blood vessel would dilate. As a result, it would get in more contact with the surface of the skin, right? And as a result, it would release the heat more efficiently, right? And the dilation of this blood vessel is what we refer to as vasodilation. And this would allow more heat transfer to the surrounding and cool the body down. So that's what I have written over here. As person moves to a hot environment, vasodilation takes place and this causes the blood cells to dilate. As a result, more blood flows through the surface to the skin, hence more heat is lost. Clear? So moving on. Okay. So scientists investigate the release of sweat from human skin when a person runs at different speed. They measure the rate of sweating in liters per hour. They take measurements in two environments. The first environment is hot and the second hot and humid, and the second environment is cool and dry. The graph shows the scientist results. Okay, this is the sweating rate and this is the speed. Okay, and the solid line would mean hot and humid, and the cool line would mean dry, cool and dry, right? So from this graph, I could see one pattern clearly, that is the gradient is being constant, right? The gradient is being constant. And let me just tell you something. So what does rate means? um Basically, I think that from your chemistry you should have this knowledge that rate is going to be volume or amount or something, or a quantity divided by time, right? This is by ball definition of rate. So basically we'd be needing this in most of our chemistry questions and biology questions, right? So have a clear understanding of what this rate is, right? So moving on. give the dependent variable of this investigation. So the dependent variable of any investigation would be on the y-axis, and the independent is going to be on the x-axis. So in y-axis we have sweating rate. Sweating rate in liters per hour, or just sweating rate would work. A person ran at a speed of 220 meters per minute in hot and humid conditions. Calculate the volume of sweat released by the person in 15 time. Okay, so it runs a speed of 220 meters per second. Okay, so this is going to be 220, right? So how did I find that? So what I did was 200 minus 160 divided by how many small boxes are there, divided by 10, right? So by doing this, it would represent how much each box represents, right? One small box represents what? So I found out that one small box represents 4, right? So yeah, so if that's 4, then 200 plus 4 times how many boxes are there? 1, 2, 3, 4, 5. 5 boxes. So that would give us 20, right? So 200 plus 20, that's 220. So 220 is over here. Or what you could do is 200 plus 4, 204, 8, 12, 16, 20, 220. You could do like that, or you could do like this. If you're facing problem in these questions, then make sure to comment below. I would be more than glad to help you all with these questions. But this is a common grasp up that you have to have an idea of at this point, because this thing is going to be extremely crucial in your A levels. After you move on to your A levels, you're not going to have the leisure to count these boxes and find it out. You have to do it by default, right? Whenever you see something, the data are going to get bigger and even clumsier with all of these decimal places and everything. So whenever you will be having these data, you have to instantly find out what you have to do with this, right? So if you're facing with this problem, then make sure to comment below. So if this is 220 for hot and humid, the sweating rate is going to be this part, right? This portion of the graph, that is 1.7. There's a catch, guys. The sweating rate is in hours and the speed is in minutes, right? The units cannot be different, right? So to convert this hours into minutes, what are you going to do, right? So that is the thing. So as I said, rate is volume by time, right? You have your rate, and what you have to do is you have to divide this rate by 60. You have to divide this rate by 60, right? And multiply it with 15, and yeah, that is how you'll be getting the volume of it. And the volume is 0.43 liter. 0.43 liter. Okay, everyone. So yeah, moving on. Just give me a while, guys. So yeah, that is how you find it in liter. So yeah, sweating rate is in liters per hour. Liters per hour, which means that liter per hour, right? But over here we have it in 15 minutes. 15 minutes. So how do you convert this 15 minutes to hour? We divided it by 60, right? So that's just what I have done over here. Clear so far? So yeah, if you're a bit unclear with this, then let me know in the comment section. Let me see if I could do this in a more easier version of it. So what I could do is I have to ensure that the units are the same, right? 15 minutes and that. So what I could do is 1.7 liters per hour equals to volume by 15 divided by 60, right? So 1.7 times 15 by 60 would mean to, yeah, this is gonna be the same thing as well, 0.43 liter. So yeah, that is how you're going to do this, but make sure that both of the units are in the same. So here what I have done is I have done the same thing, right? I have cross multiplied it and I have skipped some of the steps, right? That is how you're going to solve this. Moving on, guys. Using this information from the graph, discuss the effects of increased running on sweating in the two environments. Okay. So first thing is that as the speed increases, sweating rate or sweating increases for both the environments, right? And what I could say is, as I said, that it increases in a similar manner as the gradient is almost the same over here in both of these two environments, right? And for hot and humid, it is going to be more. Sweating is going to be more, right? Sweating is going to be more for hot and humid. So that's what I have written over here. Let's just take a look at it. With increased running speed, sweating increases. Sweating increases as well for both environments, as sweating cools the body down. More sweat is produced in hot and humid condition. Sweat evaporates more easily in drier condition. This is because diffusion gradient is greater in drier conditions. So what does this mean? I'll be explaining this in a while. Increased sweating rate is similar for both conditions as the slope is similar. Now, suppose this is the surface of the skin of a person, and these are water droplets, right? Now, if the environment has a lot of this water present, or if the environment is humid, now would this sweat diffuse into this air or evaporate into this air easily? No, right? Because what? Diffusion states that it needs to go from higher concentration to lower concentration, and if the water present over here is in higher concentration than this, then then the diffusion gradient is going to be less, right? And for that reason, I mean, if you ever had the experience to visit a humid or a tropical climate country, or a rainforest, you see that you'd be constantly sweating, right? You'd be constantly like you'd be feel your body would be filled with water because of that, because the sweat cannot evaporate much easily, right? So yeah, moving on to question 5. So this thing is going to be about a passage about cloning, right? So before we get into anything, let me just say something, right? So how do you clone something? So suppose this is the sheep that you want to clone. So what you're going to do is you're going to take a body cell from it or from a mammary gland of this sheep, and what you're going to do is you're going to extract the DNA. You're going to take that, but what the DNA is found? The DNA is found in the nucleus. So it basically means that you're going to you are taking the nucleus out of it, right? And what you're going to do is you're going to take a second female or any other sheep, right? And you're going to take an egg cell of it, and what you're going to do is you're going to remove the nucleus of this cell, right? You're going to remove the nucleus of this unfertilized egg cell. Now what you're going to do is you're going to use an electric current to fuse this DNA to this cell and from this one, right? And once you have this, you're going to place this inside a surrogate mother. Inside what? Inside a surrogate mother. But where of the surrogate mother? In the uterus, obviously, of a surrogate mother, right? So yeah, that is what I've written over here. So cloning involves taking the nucleus of a diploid body cell from the sheep to be cloned. The structure is then placed in an egg cell that has its nucleus removed, or you could say enucleated cell. Electricity is used to uh, help the cell to divide by mitosis. This then develops into an embryo, which is then placed inside the uterus of an unrelated female, and the unrelated female is what we refer to as surrogate mother. The unrelated female is what we refer to as surrogate mother. Clear, everyone? So yeah, moving on to the next question. Okay. Using recommended amount of fertilizers on a crop yield increases the crop yield. On a crop increases the crop yield. Okay. Some farmers think that adding extra fertilizers obviously will give a greater crop yield. Give a reason why extra fertilizer does not result in a greater crop yield. So first of all, the extra fertilizers would not be absorbed by the plants or would not be absorbed by the crops. And if it is not absorbed by the crops, then how is it gonna increase the yield of it, right? Because nothing in excess is good, right? Everything in excess is gonna be harmful at some point. So yeah, extra fertilizer is not absorbed by the crop. Some farmers apply more nitrogen and more water to their fields than are needed, okay? Explain the biological consequences, okay, of applying more nitrogen fertilizer. So this thing is gonna be related with eutrophication and everything, right? So whenever the person is adding excess fertilizer to a particular soil or something like that, there is always a risk of leaching, right? So what is leaching? Leaching basically means, suppose this is soil, this is the soil, and this is the fertilizer over here, right? Now if there's some source of a water, like from a rain or river, these are going to be washed away and gets into a lake, right? Now as the fertilizer is washed away into a lake or a nearby water body, what would happen is that the algae the algae in these water bodies would start to have these fertilizers and would start to grow rapidly. algae would have that fertilizer and grow rapidly. And once algae grow rapidly, that is what we refer to as algae bloom. Okay, wait, let me just check. There it is, algae bloom or not. I forgot exactly. Sorry for oh yeah, there's gonna be algae bloom. Algae bloom, because I have been teaching biochem maths both for IGCSE and A levels and A2s. So it kind of gets messed up at some point. So yeah, sorry for that. That's algae bloom, right? Now, this is the surface of water, right? Now this algae would start to grow. Now there's a clear consequence you could see. That is, light cannot enter the surface of this water because the algae is blocking the surface. So this is the surface and you have a layer filled with algae, right? So this layers is impenetrable and it is blocking light from reaching the surface of the water, right? And as a result, the plants in that cannot carry out photosynthesis, right? Aquatic plants cannot carry out photosynthesis. And the result of photosynthesis is oxygen, right? And this is going to decrease the levels of oxygen that is dissolved inside water or inside that water body. Another thing is that this algae this algae is going to die at some point of time, right? And when they are going to die, they are going to be decomposed by the decomposers, right? And when the decomposers decomposes this algae, they are going to use up oxygen. Decomposers are going to use up oxygen to decompose this algae, right? So this would cause the oxygen levels in the water to fall further. And when these oxygen levels fall further, the fishes and other aquatic organisms or aquatic plants are going to suffer from shortage of oxygen, and eventually this would lead to the death of it, right? So that's what I've written over here. Addition of excess fertilizers and water can cause leaching to take place. This causes eutrophication and algae bloom to take place. This covers the surface of the water and blocks light from reaching the lower parts of the water body. As a result, less photosynthesis takes place in aquatic plants and results in death of aquatic plants and algae, obviously. Also, as the algae dies, it is decomposed by the decomposer, which further decreases the oxygen levels. As a result, fishes suffers from shortage of oxygen and eventually dies. So yeah, guys, moving on to the next part of this question. Atmosphere can be polluted by greenhouse gases such as carbon monoxide released from the industry. Describe the effects of carbon monoxide on the human body. So you have your hemoglobin, right? You have your, suppose this is the red blood cell and this is the hemoglobin, right? Hemoglobin. Now this hemoglobin binds with oxygen to form oxyhemoglobin. And what is the purpose of this oxyhemoglobin? It transports oxygen to your body cells. Clear? Now, whenever there is carbon monoxide, it has a stronger affinity for this hemoglobin and binds with this hemoglobin instead of oxygen. So what it does is carbon monoxide has a higher attraction for a hemoglobin, this oxygen, and binds with this hemoglobin instead of this oxygen to form carboxyhemoglobin. As a result, the body cell of the person gets less oxygen, and less oxygen would mean that less aerobic respiration taking place, right? Because in aerobic respiration you would need oxygen, right? And if there is a shortage of oxygen or if there is less oxygen, it would mean that less aerobic respiration takes place, and it would mean that less energy or ATP is generated, right? Okay, so that's what I've written over here. Carbon monoxide binds with hemoglobin instead of oxygen to form carboxyhemoglobin. This decreases the affinity of hemoglobin for oxygen, and as a result, less respiration occurs. Which gas is not a greenhouse gas? Okay, let's just take a look at the option. Carbon dioxide, obviously. Methane, obviously. Nitrous oxide or NOx, NOx is obviously one of it. However, nitrogen isn't a greenhouse gas. So guys, that brings us to the end of the paper. If you find this to be helpful, then make sure to like, subscribe, and comment. And if you're facing any problems with any of these questions, then feel free to ask me in the comment section. I will be more than glad to respond to you all. Have a great day, everyone. Thank you for your time.